Hello. This lecture we are looking parasites in and near aquatic habitats. We will focus on lotic habitats, which means flowing waters. But before we get to parasites, let's look at the characteristics these habitats have. Lotic freshwater habitats are naturally rivers and streams, but there are also artificial ones, ditches and channels. We will focus on rivers and streams. Let's start from more far away and then look at these habitats more closely. The keywords are connected network, dynamic, water flow in one direction, and strong connectivity with the riparian areas. When we look at the map, we can see that rivers and streams are forming connected networks. There are small headwater streams that flow to bigger rivers. The flow goes downstream, so is in one direction, which is important if we consider the movement of different particles and also the species that live in these habitats. If not living things, for example sediments, and many not so good swimmers or flyers, they will move downstream or stay at one place. But better swimmers, for example fish, and flyers, for example emerging aquatic invertebrates, they can migrate upstream. Nowadays connectivity of the riverine network can also be fragmented by human-made dams. Next 3 slides are a bit out of the box exercises, but I think they are also important if we think about how parasites move between terrestrial and aquatic habitats, and also how they can impact species assemblages and ecosystem processes. Rivers and streams are strongly impacted by the landscape that surrounds them, but rivers and streams themselves are also impacting the landscapes that they are flowing through. Hydrologically, we could say that the river is wider than the channel where the water is flowing. Water level is dynamic and impacts also the groundwater movements. Lateral connectivity means how water impacts the habitats to the side of the river, for example flooded floodplains. Vertical connectivity, on the other hand, means how river bottom is connected to the hyporheic zone that is below the riverbed. There are movements of different species in both of these mentioned directions. Nowadays, lateral and vertical connectivity can be hindered by human-made channelization, artificial riverbeds, or excessive fine sediment accumulation. Also biologically, rivers and streams are wider than the channel where the water flows. The median stream channel width in this study was 3.5 meters, but invertebrate species that originate from the stream migrate in their adult stage more far away to the riparian area. The distance is different between taxonomic groups, but mean distance where half of the invertebrates were found was 1.5 meters, and 10% of the invertebrates were even found 550 meters away from the stream. The numbers or the abundances were compared with the abundances that were on the stream. So biologically, a 3.5 meter river can be more than 500 meters wide, in the sense that the species that lived and foraged in the river, they are migrating so far away. When we know that species movement between terrestrial and aquatic habitats when we know that species move between terrestrial and aquatic habitats, then we could also assume that this is important considering food webs. It has been found that, for example, up to half of drift-feeding fishes' annual energy comes from terrestrial invertebrates that are falling to the stream. The example of the drift-feeding fish is trout. The other way around, the scientists found that 25 to 100% of energy or carbon of the riparian consumers, for example birds, bats, lizards, spiders, originates from invertebrates that lived in stream as larvae and emigrated from there as adults. When we consider all we looked at previous slides, then I think it is important to think what role and impacts can parasites have in this system, and also how these reciprocal movements and dynamic environments are impacting the parasites themselves. Now, if we have looked more closely at the functioning of freshwater habitats and adjacent areas, it's time to focus more on parasites and look for some examples. Firstly, about some trends from 2010 article. During this time it was reported that in the last 40 years freshwater parasite infections increased in the groups of amphibians, fishes and crayfishes. During the same time infections in mollusks decreased and no change was reported in freshwater reptiles, birds and mammals. If you're interested which parasite group dominated different freshwater taxonomic groups, then you could press pause and read shortly the next list. In the same article, the researchers described that freshwater environments can function as focal points for pathogens. The reasons may be because fresh water is necessary for organism survival, and there is a lot of it in these habitats. Also, there are many vectors during aquatic invertebrate life histories, and they are often intermediate hosts for the parasites. Often aquatic and terrestrial species near water concentrate in aggregations, so they are gathering there, which offers possibilities for parasite transmissions. Freshwaters are also one of the world's most changed habitats, which can affect species interactions and disease course. Freshwater habitats are disproportionately biodiverse compared to the area of the freshwater habitats, so they can be considered as biodiversity hotspots. If we consider the previously mentioned statements, then maybe there are questions raising if these freshwater habitats can also be hotspots for parasites. Now let's look at some examples. The researchers looked for 5 reservoirs that these are human-made lakes behind dams. And they investigated them and found out that 6 snail species hosted 36 trematode species. The trematode species are parasites. The figure on the slide shows transmission pathways of the trematode species, and the middle boxes are the second intermediate hosts or encystment in the aquatic environment, and the top boxes of the figure are definitive host groups. The lines are trematode species utilizing individual transmission pathways, and the thickness of the lines are proportional to the number of trematode species utilizing each pathway. The dotted lines show direct infection of the definitive host, so then there is no intermediate host or encystment. The abbreviations at the bottom of the figure are different reservoir lakes that were investigated. And what was interesting is that whereas two most abundant snail species hosted for almost 90% of the trematode diversity and also spatially and temporally stable parasite communities. That's why stable host populations, which are abundant and stable in space and time, are important for parasite movements and diversity. I think it's comparable with the more known concept that bigger habitats support higher biodiversity and abundances within species. It's also a good figure to analyze or to visualize the hidden connections between different taxonomic groups in an ecosystem, and here in aquatic ecosystem. So now next 3 slides are about parasites that mostly are not lethal and probably doesn't cause so much problems to the host species. They are also quite big. If you look at the parasite scale, that means that mostly or quite often it is possible to see them with the naked eye or by using the microscope, but not making it so big. So all the big freshwater mussels in Estonia have parasitic larvae called clochidia. That means every mussel that we find in lake or in a river has lived in the beginning of its life as a parasite. And the big mussels here mean that the mussel grows, when it's adult, it grows larger than 3 centimeters. There are also 2 mussel species in Estonia that prefer only flowing waters, and together all the big freshwater mussel species in Estonia, there are 7 species together. The 2 ones that prefer flowing waters, they are thick-shelled river mussel and freshwater pearl mussel. Some of the mussels' larvae have hooks and some not. For example, thick-shelled river mussel larvae has hooks, and the freshwater pearl mussel doesn't have hooks. And that's probably why the thick-shelled river mussel can live also on the fins and the gills of the fish, so it attaches with the hooks to the fins of the fish. But freshwater pearl mussel only lives on the gills of the host fish. Thick-shelled river mussel has several host fishes, for example European minnow, bullfish, and perch, and also others. Whereas freshwater pearl mussel can be or live parasite mostly only on brown trout and Atlantic salmon. And depending on the water body, there are usually host fishes that are more abundant and also more suitable to the thick-shelled river mussel or to the freshwater pearl mussel in this specific water body. Now you can look at the video of the freshwater pearl mussel clochidia snapping their shells together, which is the same reaction when the larva happens to get on the gills of the fish, and then it snaps the shells together and attaches itself to the gills. And on the other side there is 1 minute video about thick-shelled river mussel behavior. So when it's time to release the clochidia to the water, then quite often the thick-shelled river mussel uses its leg and roams to the side of the stream or river, and then it spurts or like shoots the water up to 1 meter in the direction of the middle of the river, and it has been thought, the scientists are thinking that it's so that it like attracts the host fish to the place where the clostridia is, and so the host fish gets infected. But it has been also researched that some of the clostridia gets eaten by the host fish, and also quite a lot of the clostridia never fall to the fish, it just falls to the river bottom or somewhere else. So, next slide. On this slide you can see young freshwater pearl mussels who did recently fall from the gills of infected brown trout. They already have 1 leg, which is used to move and forage. It's also possible to see life cycle of the mussel on the slide, and also adult mussels at the river bottom. Currently it is known that this species, freshwater pearl mussel, lives in only 1 river in Estonia. But the mussels are not the only ones who want, in question mark, or who wants their young ones cared by other species. There is a fish species called European bitterling that inserts its eggs between the gills of the freshwater mussels. Since the mussels are filter feeding, then the male bitterling fish is releasing their sperm in the water column near the mussel, and this way the mussel itself is taking care of the fertilization of the fish eggs. And researchers found out that in places where co-evolution between mussels and bitterling has been taking place significantly longer, it's 2 million years versus about 160 years, then in the place where it has been, the co-evolution has been taking place about 2 million years, the mussels have better defense mechanism against the fish spawning. So our last topic looks at maybe a bigger picture about how parasites are linking terrestrial and aquatic habitats, and how they may impact species assemblages, food webs, and even impact the ecosystem processes. Here you can see the life cycle of parasitic nematomorpha, also known as the horsehair worm. As adults they are free living in aquatic habitat. After mating there will be larvae that gets into aquatic invertebrate. There it then cysts, and the cyst survives also the metamorphosis of the aquatic invertebrate. Now, when the aquatic invertebrate emerges from the water and flies to the terrestrial habitat, then it may get eaten by the cricket, and then the cricket gets infected. The parasite cysts in the cricket developed worms that live in hemicoil of the host, and the parasite forces the cricket to go to the stream. Now, it's quite interesting because the parasite forced the cricket to go to the stream where it is vulnerable to aquatic predators, for example fish and frogs. And in their experiment of these researchers, adding infected crickets at natural levels was an important food source to the fish that is drift feeding. That means that it feeds from the insects that are falling from terrestrial habitat. And that way the predation pressure on the stream benthic invertebrates decrease, because fish now did eat the crickets and other terrestrial invertebrates. And the biomass of stream benthic invertebrates increased more than 3 times, and also the community structure changed. And since there was more benthic invertebrates, then they forage more benthic algae, and then the biomass of benthic algae was reduced by half. And if they wanted to look at ecosystem processes, then quite often it's used. The method is to look leaf breakdown rate, so how fast the leaves in the stream are breaking down, which is really important in the ecosystem. And when they added crickets, then the difference was significant if it was compared with 2 other treatment groups, which was not adding infected crickets and also hindering all the terrestrial invertebrates to fall to stream. So then the leaf breakdown was significantly faster when they added crickets, because then there was more benthic invertebrates that participated in breaking down the leaves. So these horsehair worm parasites had quite important impact to the terrestrial aquatic food webs, also the species assemblages and even ecosystem processes. And when we add the nature conservation point of view, then researchers have also found that intensive forest management appears to disrupt the life cycle of the horsehair worm, which can reduce the terrestrial subsidy to trout streams, and so have other impacts also to stream biota. Now, to sum up, parasites are important and connecting factors of lotic freshwater ecosystems, but the impact of them can often be hard to see because they are small and connected with many vectors. So what do you think? Maybe it would be worth a thought to reconsider the negative image of parasites in aquatic and also other ecosystems.